So this is part 2 of IB Bio 11.1 antibody production and vaccination. So last time in part 1, we spoke about these phagocytes, the white blood cells that go through phagocytosis and eat the bacterium. You can also call these macrophages. They eat the bacterium or the pathogen. They would be taking the antigen, which is noted as the little red dot there, taking the antigen and then presenting it on its surface, on the surface of the cell of the phagocyte, presents it to the helper T cell. The helper T cells then take, they have a specific receptor that actually matches up with the antigen and recognizes the antigen. And so that is part of why this is considered specific immunity, because there's a specific helper T cell that specifically matches its receptor to the antigen that is presented to it. And remember, the antigen was on the surface of the pathogen. Okay, so then it is now activated, the helper T cell. Then the helper T cell has to do the second step of this, all of this, which is to activate the B lymphocyte or the B cell. So it comes over, and this is showing it activating the B cell over here. And notice that the B cell also has its own specific receptor that also matches with the specific antigen on that pathogen. And so there are thousands of different helper T cells and thousands of different B cells. And then it just takes one specific one to match its receptor properly to the antigen. Okay, then now we're at the point that the B cell is activated. The B cell goes on and it does divide into two types of cells. Okay, so it makes clones of itself. We call it clonal selection, and it forms plasma cells and memory cells. The memory cells are very important because that is what confers or creates immunity within the body. They remain around, and we'll talk about that in a little bit. They remain around in case your body gets reintroduced to that same pathogen and has to reactivate and form more of the plasma cells, which then would be producing those antibodies that attach to the antigen on the pathogen and renders the pathogen useless or marks it for destruction. This is an image of an antibody. We talked about all of the different ways that the antibodies do destroy or render the pathogen useless. So go ahead and look back at my part 1 video if you want a review of that. Now we're moving on to vaccines. So what are vaccines? They do contain antigens, and this is how vaccines function. It's basically like an artificial way of causing specific immunity. So for example, this whole process that we just talked about, it causes that to happen because it introduces the antigen into the body. And so there are several different forms of vaccines, and even now there's some vaccines that are DNA or RNA vaccines as well. Vaccines are either attenuated, which means weakened or inactivated, forms of viruses. So it wouldn't be the full functioning virus that would cause a person to get sick, but it would just be a weakened or inactivated form of the virus, just so that we can introduce the antigen to somebody's body so that we can have the specific immune response. You can also have weakened toxins, which would be a bacterial vaccine, which does exist as well. But usually we think of vaccines for viruses, not for bacteria. Then we've also got subunit vaccines, which could just be the antigen or part of the pathogen containing or carrying the antigen. So again, we're just introducing the antigen to the body so that it has that specific immune response. So the way that vaccines work in terms of antibody response is that first there's a primary immune response in response to the antigens that were introduced into the body after the vaccine. Okay, and as a result of that, there are some memory cells that were produced, just like when we were talking about here with specific immune response. Remember that the B cells divide into plasma cells and memory cells. And so those memory cells are what is being referred to here. Those memory cells do remain even after the plasma cells, are gone and the antibodies that were produced are much less. And then the secondary response is actually stronger and more rapid than the primary response because your immune system essentially already is prepared to fight off that pathogen. And so it's going to ramp up into full antibody response and increase antibody production. And then, of course, the pathogen would hopefully be destroyed and the antibodies would go back down again. Okay, so just a reminder slide of what you could potentially be asked on an IB exam is explain the specific immune response starting with a pathogen entering the bloodstream. And so you would just go through this response here. I would say for this part, helper T cell stimulates appropriate B cell. That would mean the helper T cell is activating the appropriate B cell. B cells producing clones, or you can say it's dividing and making copies of itself to become plasma cells, or really it's and memory cells. Another thing about this is the macrophage takes on the antigen, so you don't have to mention epitope. I wouldn't say that you have to mention epitope. You can just say it takes on the antigen and then it presents the antigen to the T cells. For this statement, you're supposed to know that smallpox was the first infectious disease of humans to have been eradicated by vaccination. Okay, the other thing that you're going to want to know is the nature of science statement about considering ethical implications of research. For example, with the Dr. Jenner, who tested his vaccine for smallpox on a child. So he actually deliberately infected an 8-year-old boy with cowpox. And so cowpox is a similar disease to smallpox. He actually took some of the cowpox virus from the pox and blisters of a milkmaid. So this woman had cowpox and that had the pox or blisters that form on the skin. So he actually took the virus from that and then infected the boy, the 8-year-old boy, with cowpox on purpose. And then he proceeded to attempt to infect the boy with smallpox. He found that the boy was immune to smallpox because cowpox is a very similar disease, so it actually gave him immunity to smallpox. While this did result in eradication of smallpox and ended up being, a great thing because it was the first infectious disease to be eradicated by a vaccine, it was extremely unethical for him to be testing a vaccine on an 8-year-old child. All right, this statement is just about how pathogens can be species specific, although others can cross species barriers. So some pathogens, such as polio, measles, and syphilis, which are all viruses, can actually only affect humans. However, there are others like the flu, Ebola, Salmonella, that can actually be transmitted between humans and other species. For this statement, I want you to read it very carefully because it can be slightly confusing. You may have to practice this concept a few times before you really get it. So the fusion of a tumor cell with an antibody-producing plasma cell creates a hybridoma cell. Okay, monoclonal antibodies are produced by hybridoma cells. What in the world does this mean? So we've got a melanoma cell here, which is a type of tumor cell. Okay, so we're fusing the tumor cell with an antibody producing plasma cell. So if you remember back when we talked about specific immunity, over here, we've got once the B cells are activated by the helper T cells, they divide into plasma cells and memory cells. What do these plasma cells do? They produce antibodies, okay? So this is essentially a technique that scientists have learned to be able to do to produce antibodies. That's the end goal of all of this, is to sort of artificially produce antibodies in the lab so that we can use it for therapeutic purposes. So how do we do that? Well, we fuse a tumor cell, such as a melanoma cell, with that B cell, the plasma cell that the B cell turned into, that's producing antibodies. So we've fused them together. They are now essentially one cell. Then, what do tumor cells do? Why are tumor cells a problem? Well, because they replicate, right? They replicate over and over and over again. And so that's what scientists are taking advantage of, that characteristic of the tumor cell, and taking advantage of the fact that the B plasma cell is producing antibodies and fusing them together so that they can make multiple copies and clones of themselves very easily in the lab. And then we have unlimited amounts of antibodies that are produced. So here's a summary of all of the steps in order to do this process. Okay, we call those antibodies that end up being produced monoclonal antibodies because they are just clones of that fused cell. And by the way, we call that fusion of the tumor cell and the B cell a hybridoma cell. Okay, and so that's also in the statement right there. So if you're ever asked about hybridoma cells, it is a fused tumor cell and a B plasma cell that is producing the antibodies. Here we have a summary of the steps again. So first, we've injected the antigen or a vaccine into the animal, such as the mouse. The mouse starts producing these plasma cells, right, because of its specific immune response, and it's producing antibodies. The plasma cell is producing antibodies to fight off this pathogen or antigen. Okay, that plasma cell gets fused with the tumor cell. We now have what's known as a hybridoma cell. And then that hybridoma cell is producing an endless supply of monoclonal antibodies so that we can use it for therapeutic treatment of, for example, an emergency treatment of rabies. If somebody had rabies and we needed to give them tons of antibodies as soon as possible, well, we can artificially make those antibodies in the lab using this process, and then inject those antibodies into the person, and they can start fighting off the rabies virus. For this statement, monoclonal antibodies to HCG are used in pregnancy test kits. So, I believe all that you really need to know as far as the IB exam is just that the monoclonal antibodies that we spoke about in the previous slide, right, that are produced by fusing the plasma cell to the tumor cell to make the hybridoma cell, right, those monoclonal antibodies that are produced, so another function of those antibodies is actually for pregnancy test kits. And how does this actually all work? This animation is very helpful in explaining exactly how they work. It does get pretty complex and complicated. I will go through this and talk about it a little bit. That being said, I don't think that the IB exam will get into so much of the specifics of how all of this works. But remember, the main idea here is that we're producing those antibodies in the lab, the monoclonal antibodies, in order for therapies such as with rabies, as I explained before, but then this is another application of using those antibodies.
A urine sample can be tested to determine whether a woman is pregnant. This test is based on the ability of antibodies placed on commercially prepared test strips to bind to specific molecules in the urine. The number of colored bands on a test strip determines whether the result is positive or negative. The urine of pregnant women contains a hormone called human chorionic gonadotropin, or HCG. HCG is produced by the developing placenta and has important functions in pregnancy. However, some of this hormone is also excreted in the woman's urine. Women who are not pregnant do not produce HCG. An HCG test strip can detect low levels of HCG in the urine. The strip is made of a capillary membrane that draws urine by capillary action along its length. The test strips contain 3 zones of function, here labeled R, T, and C, to indicate a reaction zone, test zone, and control zone. The reaction zone contains a type of antibody called anti-HCG antibody. These are mouse monoclonal antibodies which recognize a single molecular feature or epitope on HCG.
Okay, so these are those monoclonal antibodies that were produced in the lab using the hybridoma cell, and they are attached to this reaction zone right here. They can detect and bind to HCG, that hormone that is produced from women that are pregnant. Women who are not pregnant do not produce that hormone.
Molecules. The letter E signifies that these mouse antibodies have been artificially linked to an enzyme. The test zone contains a polyclonal mixture of antibodies, which recognize a variety of epitopes on HCG molecules. This zone also contains dye molecules, which act as substrates for the enzymes currently in the reaction zone.
These are other types of antibodies that are produced in the lab that also can detect the presence of and bind to HCG.
The enzymes in the reaction zone can catalyze a color reaction with the dye substrate. The control zone contains the dye substrates, as well as a preparation of antibodies. These antibodies, called anti-mouse antibodies, can recognize epitopes on the mouse monoclonal antibodies that are currently in the reaction zone. The urine of a pregnant woman yields a positive test result. To determine this result, the test strip is dipped in the urine sample. The sample, which contains HCG, is drawn up the strip by capillary action and first arrives at the reaction zone, which contains the enzyme-linked anti-HCG antibodies. The enzyme-linked antibodies recognize parts of the HCG molecules and bind to these molecules from the urine. The complexes of HCG and antibodies, as well as the unbound antibodies, dissolve in the fluid and detach from the capillary membrane. They are then carried along the
Okay, so this is sort of starting to get into how the antibodies, the monoclonal antibodies that are produced, are actually used. They can not only bind to antigens on pathogens, as we were talking about before, but they can also bind to very specific molecules, such as the HCG hormone that's produced in the urine of a pregnant woman. And so they binded to the HCG hormone, some of them, and some of those monoclonal antibodies did remain unbound, and they're just being carried up the pregnancy test strip.
test strip by the capillary flow. The complexes and free antibodies arrive at the test zone. This zone contains the polyclonal antibodies and the dye substrate, both of which are permanently affixed to the strip. The immobilized polyclonal antibodies bind to HCG molecules in the complexes. The free antibodies continue.
So notice that if there's just the free antibodies that did not bind to HCG, then the free antibodies just keep moving along. But the antibodies that did bind to HCG do stay in the test zone because they got bound to these polyclonal antibodies that were manufactured in the lab to bind to the HCG and the monoclonal antibody.
To flow along the strip. The mouse monoclonal antibodies that escaped binding in the test zone have reached the control zone. Here, anti-mouse antibodies that are affixed to the strip bind to these monoclonal antibodies.
So this is the control zone because it should always react and catch these free antibodies that did not react with the HCG. So no matter what, This step should be happening, and those dye molecules, as you'll see later, will be activated and turn red.
In the test zone, the enzyme-linked antibodies catalyze a reaction with the dye substrate and trigger a color reaction. The same type of reaction occurs in the control zone.
So notice how on a negative pregnancy test, this test zone does not get activated and does not turn red because there was no HCG bound to the monoclonal antibodies, so there was no reaction that ended up occurring.
In a positive test, 2 of the 3 bands are colored, but in a negative test, only 1 is colored. This difference results from a lack of HCG in the woman's urine. The test begins when a strip is dipped in the sample urine. The sample migrates to the reaction zone, which contains the enzyme-linked anti-HCG antibodies. Because there are no HCG molecules, these mouse monoclonal antibodies have nothing to bind to, and therefore migrate alone up the test strip. The enzyme-linked antibodies arrive at the test zone, which contains a dye substrate and the immobilized polyclonal antibodies. Here, the enzyme-linked antibodies are not carrying HCG molecules, so the immobilized polyclonal antibodies are not able to snare them. All of the enzyme-linked antibodies continue to travel along the strip. The enzyme-linked mouse monoclonal antibodies are captured in the control zone. The control zone contains anti-mouse antibodies that bind to regions of the traveling antibodies and thereby immobilize them. The control zone is an important region that helps distinguish whether the strips are working correctly.